So welcome everyone. I'm Dr. Mike Goldberg. I'm the board president of the RYR1 Foundation. We have a lot of information to review tonight, so I want to jump right in. First, I want to welcome Dr. Paya Mahassel, Dr. Josh Todd, and Dr. Jean Marcantonio. On behalf of the entire RYR1 community, thank you for your work on the Rycal trial, and thank you for being here tonight. I thought we'd just go around and maybe each introduce ourselves. So Dr. Marcantonio, do you want to go first?
Sure. I'm Jean Marcantonio. I'm the CEO and CMO of Armgo Pharma. I was the medical monitor on this trial, and I'm here to answer questions about Armgo in general and the compound, et cetera.
Great. All right. Dr. Mahassel.
I'm Paya Mahassel. I'm an adult neuromuscular neurologist, currently an associate professor of neurology at Johns Hopkins, and I was the PI of the Rycal trial.
And PI means?
Principal investigator.
Thank you. All right. And Dr. Todd.
Josh Todd, I'm staff scientist at the National Institute of Neurological Disorders and Stroke, and I was the lead associate investigator on the Rycal trial.
Great. Well, thank you. We owe these individuals a huge debt of gratitude. They each spent countless hours preparing for this trial, performing the trial, and reviewing the data, and then ultimately publishing the recent manuscript. In addition, I think this trial is illustrative of the positive partnership between academia, where the basic science of Rycals was done, industry like Armgo Pharma, and a nonprofit organization like the RYR1 Foundation. The RYR1 Foundation provided funding for some of the basic science that provided justification for this clinical trial. And we also served as an advocacy role in which we went to the leadership at NIH and, yes, the halls of the United States Congress to ensure that this clinical trial was done. We just weren't not going to accept no for an answer. And I don't think it's an exaggeration to say that were it not for the RYR1 Foundation, this trial would not have happened. Lastly, thanks to all of you who have logged in tonight. We had over 220 people sign up for tonight's webinar, and there have been countless additional people who have posted saying they can't join us tonight, but they want to know if this is being recorded, and it is. So we have an additional large number of people who aren't here right now, but will be watching this at some later time and date. This is a meaningful number to someone like Dr. Marcantonio, the CEO of a pharmaceutical company, but it also says a lot to other researchers, clinicians, biotech and pharma, and investors that there is a large, motivated, and enthusiastic patient population that is very interested in a therapy for RYR1-related diseases. Finally, we're about to jump in and review the data from the Rycal trial. But I would argue that one of the most important takeaways from tonight is not necessarily just the data from this trial, but rather that a trial was done and it was completed. And that is no easy feat. And so that is an important piece of information, again, to researchers, clinicians, biotech and pharmaceutical companies, and investors, that a successful trial of RYR1 patients can be done. And if nothing else, if you remember nothing else from tonight about the elements of the data, just remember that is hope for the future, that a trial can be done. And we hope this is going to be the one of many future trials that occur for RYR1. So with that, we're going to jump into the questions here. And the way the format is going to work tonight is we're going to start off with just some basic questions about Rycals. We'll then move to the clinical trial, and then lastly, we'll wrap up and we'll talk about, well, what is the future of Rycal? So just to start off here with some basic questions to make sure we're all on the same page, can anyone on the panel just talk briefly about what are Rycals? What does that mean? Can just describe that drug and its mechanism of action?
So I guess I can go ahead. So Rycals are a term for a group of compounds that bind to the ryanodine receptor, either one or two or both, and that modulate the calcium response. And in this case, the drug is ARM210 or S48168, as it was previously known. And that molecule binds to a site on the ryanodine receptor and causes it to be repaired. In other words, when it is leaky, when the channel is leaky, as is the case in patients with RYR1 mutations, the drug binds to those leaky channels. And because it's binding to a site that seems to be important in the conformation of the structure of the channel, it causes the channel to be repaired. It doesn't bind to where calcium comes out. So there was a pore in the channel, a hole, if you will, in the membrane that the calcium can flow. It does not bind there. It binds elsewhere and allows that pore to be appropriately regulated. as it should be normally.
Now, Dr. Marcantonio, you said RYR1, 1 or 2, and I want to make sure people are clear on that. You aren't saying 1 receptor or 2 receptors. Can you just briefly talk about what you meant by RYR1 versus RYR2?
Right. So RYR1 is the major calcium channel in skeletal muscle. It's present in a few other areas of the brain, like in the Purkinje cells in the brain and in other places of the body, but mostly it's in skeletal muscle. And RYR2, which is present in many places, is also important because there are genetic mutations there that cause a cardiac disease, a very bad cardiac arrhythmia. And we, in fact, have a phase 2 trial in RYR2 mutation patients ongoing. Our drugs bind to equilibrium them well. However, our drug also prefers the leaky channel over the wild type channel. So in someone with an RYR1 leak, it will bind preferentially there and not to RYR2 so much. And someone with an RYR2 leak, it will bind to RYR2 preferably and not to RYR1 much. Now, there are some diseases we may get into that have both. Duchenne's muscular dystrophy in later stages is one. Heart disease, cardiac failure, congestive heart failure is another. And in those cases, the drug can bind to both RYR1 and RYR2 because they're both leaky.
Right. All right, great. Thank you for that description. Can you briefly summarize the research that had been done on Rycals as it relates to skeletal muscle that led up to the clinical trial?
Sure. So much of the work was done by our founder, Andy Marks, who's the chair of physiology and biophysics at Columbia University School of Medicine. And Andy's work, Andy was the first person to actually clone the cDNA for RYR1 many years ago. He studied it extensively in animals. And he also, together with Katie Millar at the NIH, biopsied RYR1 patients and took their muscle biopsies, was able to show that the drug given ex vivo, that is after the biopsies had been taken out of the body, could actually repair the channels of 17 or 18 different mutations. So if the channel is leaky, our drug can repair it. If the mutation causes the leaky channel, that will be repaired. It seems to be independent of where exactly the mutation is, as long as it causes a leaky channel. That's the fundamental work there. There's some other work that's been involved, but that work, I think, gave us promise that if we got enough drug to the muscle, that we could recapitulate that result in vitro of fixing the channel.
Right. And that study of those 17 muscle biopsy samples taken from patients with RYR1, it's referenced in the most recent article about the clinical trial. And just a question about that, because I think some people may have, if people notice that, may have a question on this. That study, and in the article they referred to S107 as the Rycal, and yet this paper, this clinical trial, refers to S48168 or ARM210. Can you just clarify those different terms and what they mean and the similarities and differences between them?
Sure. So S107 is a tool compound. It's a compound that Dr. Marks and we often use to do animal testing or in vitro testing. Unfortunately, S107 is not suitable for dosage in people for a number of reasons. Its half-life and its profile, it's not as specific as ARM210, which has a very nice long half-life, can be given once a day by a pill. S107 cannot. But for tool purposes and for many of the earlier studies, S107 was used as a Rycal in animal studies and has been a very effective one at that. It also has the advantage in studies to be brain penetrant, which ARM210 is not, but we don't need ARM210 to be brain penetrant to treat RYR1 because the bulk of it is in the skeletal muscle.
And one last thing before we just move on, just to clarify, we're talking about RYR1, and that can have several different meanings. We can talk about the RYR1 gene, and we've been largely talking about the RYR1 receptor, the calcium channel. And just to make sure everyone's on the same page, for those patients who have RYR1 related disease, they will have a mutation or multiple mutations in their RYR1 gene, which will have a negative effect or a harmful or deleterious effect on the RYR1 receptor. And this drug, the Rycals, acts at the level of the receptor. We're not talking about gene therapy or gene editing today. Okay, so let's move on to the actual trial. And this will be mainly for Dr. Mahassel and Dr. Todd. So just starting off really basic, let's just start off with the title. In the title, you talk about phase 1, you talk about quote open label, and you talk about dose escalation. Can you briefly summarize for everyone what those terms mean and what their significance is?
I can take
Sure. Josh, do you want to take it or I?
Sure, why not? So phase 1 is really referring to an early stage of drug development where the focus is primarily on safety. And that was the focus of this trial of ARM210 in RYR1. In terms of open label, this really means that both the participant and the study team are aware of what the participant is receiving. For example, there was no placebo involved in this trial. And in terms of dose escalation, what we're referring to here is that more than one dose was tested in a stepwise manner. So for example, in this study, you'll see that participants initially enrolled received 120 milligrams of ARM210 daily for 28 days. The data was then reviewed by an independent board, and then the remaining participants received 200 milligrams daily for 28 days.
Right. And can you talk about, you mentioned a placebo wasn't used in the open label. Can you, just to make sure everyone's on the same page, what is a placebo and what would be the significance of not using a placebo versus using a placebo in a study like this?
Yeah, so placebo is effectively a dummy tablet, if you will, that does not contain any active product. So it would be sort of like a sugar tablet that doesn't contain any ARM210. The importance of using placebos in later stage development of clinical trials is that it can help rule out bias. But it wasn't necessarily appropriate at this early stage of clinical development where the focus was on safety as the primary endpoint of the study.
Okay, great. Thank you. And we're going to get to that concept of the primary endpoint being safety in a second. Just want to lay a little more groundwork here in the manuscript. Dr. Mahassel, you talk about the participants were both monoallelic and biallelic. Can you explain what that means in some terms that may be interchangeable with those terms?
Sure, yeah. So I think most people are aware that RYR1 is complicated, and specifically the genetics of it are also not always straightforward. So there are some individuals who have the RYR1 related disease when they have a single mutation in one of the copies of the gene. So we refer to those individuals as monoallelic, meaning just one of the copies of the gene has a mutation. And there's some individuals who have mutations in both copies of their RYR1 gene. We refer to that group of individuals as biallelic. So they have both copies affected. It's kind of not completely, but similar to using the words dominant instead of monoallelic and recessive instead of biallelic. But it's monoallelic and biallelic are probably more inclusive of all the different scenarios that can give you. So that's why we chose those terms to refer to these two different genetic situations.
Right. So, okay, that's helpful. And you and I actually talked about this the other day. And I think for people who see that or are still concerned, as you mentioned, dominant and recessive, there's a lot of overlap. They may not be completely one-to-one, but it's probably fair or safe enough if you're thinking about dominant or recessive to use those terms. But the more precise terms are in fact monoallelic and biallelic. Okay, so Dr. Todd, you mentioned a couple minutes ago, you said the primary endpoint was safety and tolerability. Can you explain what that means? Because I think that's a really key point here. Because I think if we're thinking about efficacy, I think most people think about a clinical trial. Did the drug work? And in this case, we may be talking about, well, did it make someone stronger or did it reduce their fatigue? And so, can you describe what the term primary endpoint means, and then in the context of this study, safety and tolerability, and what that means or what it doesn't mean?
Sure. So the primary endpoint is really the question that the study is designed to answer. Like I said, at this early stage of development, the focus was on safety as the primary endpoint. In clinical trials, safety is assessed by looking at adverse events. And these are medical events that could be For example, a clinically significant lab abnormality would be considered an adverse event. The PI, principal investigator, is responsible for determining whether that particular event is related to the investigational product, in this case ARM210, or unrelated to the investigational product. Additionally, each adverse event is graded in terms of severity: mild, moderate, severe, life-threatening, and death. In this particular trial, there were 27 adverse events recorded. 24 were mild and 3 were moderate. However, all moderate AEs were deemed unrelated to ARM210 and had self-resolved by the time of study completion. So that gets at the safety piece. Tolerability is really looking at compliance to the intervention. That's probably the best readout for that. And what we saw here is that participants actually reported 100% compliance with ARM210. And we assessed that by taking pill counts at the middle of the study and at the end of the study.
Got it. So people who were enrolled, there were no significant safety events. In other words, if someone slips and falls while taking the drug, and they hit their head, and it's just and they have significant, let's say, head trauma, but you were to assess it has nothing to do with the drug. It's the fact that they slipped on a banana peel on the floor. That's what we're talking about here. That you could have an adverse event, but you found that in this trial, none of the significant adverse events had anything to do with the drug. Is that correct?
That's absolutely correct.
And then on the tolerability, people were able to take the drugs. They didn't find it to be too burdensome to take all the pills. They were able to follow a schedule. And so there were no issues there. Is that correct?
Exactly.
And then you talked earlier about the low dose group and the high dose group, that the low dose group, which were the first few patients, were on 120 milligrams per day, and the high dose group were on 200 milligrams per day. Did that have any bearing on safety and tolerability?
It did not.
Okay, well, so that's great news. All right, so moving on to a little bit beyond safety and tolerability, the article talks about target engagement. What does that mean? And could you maybe tie that back to that original study on the muscle biopsies? And that would be for Dr. Mahassel or Dr. Todd.
Sure. So target engagement is something that during drug development is really important because what it really means is that whether the drug that you are administering during the study is going to engage with where it's designed to go. So in this case, does ARM210 engage with the ryanodine receptor in the skeletal muscle tissue when it's taken by mouth, at the doses? Now, it's not always easy to prove target engagement. It is a difficult task, and there are many drugs out there which are approved for different indications and have never really shown definitive target engagement. But for rare disease, and especially drug development in the rare disease space, target engagement can really accelerate this process. So in this trial, we knew that we were going to enroll only a few individuals. This is a rare condition. We weren't going to have access to hundreds of people to be in this trial. And so we wanted to look at target engagement, but as an exploratory outcome measure. What does that mean? Exploratory meaning that we're going to look at it, but it's not the main thing that we're trying to prove as part of this study. And so we used some of the muscle biopsy samples that we obtained during this trial to kind of get a sense whether we have that or not. We have that target engagement or not. Understanding that given the small number of samples, we were not necessarily poised to prove or disprove this. And so as you pointed out in this article, we talk about the target engagement assays that we use, and we show some of the data that's there. But at this point, based on the data that we obtained, we couldn't say one way or another we had target engagement with ARM210. And that goes back to what Dr. Marc Antonio was talking about earlier. When we had the luxury of doing the before this clinical trial, when the muscle biopsy samples were in a test tube and you were adding the drug to it, it's a little bit easier to kind of assess the target engagement. And that study was pretty straightforward, more straightforward and pretty convincing. But when you give the drug by mouth, to a participant and then do a muscle biopsy, it becomes a little bit more challenging. And so that's what we did in the study here.
Got it.
So Mike, I would just add one thing. Target engagement in drug development is most important to establish if the drug does not have efficacy. So essentially, when I worked previously, I used target engagement often to kill mechanisms because you would show that the drug did indeed gauge the target, but did not accomplish the clinical goal. So if you have efficacy, as Payam has pointed out, in cases where there isn't target engagement, it's not a big deal because you're getting efficacy. But when you don't have efficacy in a late stage trial, having the target engagement means you've reached what you're supposed to, yet the mechanism didn't work. In our case, I think we're finding out that the mechanism is working, but that's another yet to happen. So,
and then, okay, just one quick follow-up on this point, Dr. Mahassel. So a little bit unclear about target engagement, small sample size, some technical issues. But you did mention in the article that there was tissue penetration. So can you talk about tissue penetration versus target engagement and what your thoughts on that are?
Sure. So, when you take any pill or drug, by mouth, it goes through the metabolism of the body, it gets absorbed, gets through the liver, and then it needs to go where it's supposed to go. So if you're taking a drug, for example, for pneumonia, it needs to get to the lungs where the bacteria are and do its work. So the issue of its distribution to the tissue that you're trying to target is what we meant by tissue penetration. So does the drug, when taken by mouth, get to the muscle tissue? And we were able to measure the drug in the muscle tissue of the participants in this trial that had that portion of the analysis done. So the drug does get to the muscle. Target engagement is sort of a couple of steps after that. So the drug has to get there, but then it also has to get to its target and have the effect that we thought it was going to have on the target. And so because it has these additional steps, it's not easy to prove. It's easier to measure a drug, in a tissue compartment than to show that it engaged, its target and had the effect that we expected.
Okay, great. All right, so I want to move on a little bit to away from safety and tolerability to, I think the term used is preliminary efficacy. And just to be clear, just to reiterate, given the size of this trial, the small number of people, the overall goal, we talked about phase 1, we talked about safety and tolerability. The goal of this study was not necessarily to prove efficacy, that is, improving strength or functional outcomes or so on and so forth. But you did what you were talking about, preliminary efficacy. So can you talk about what that term means, preliminary efficacy, how you assessed it, and what your takeaways were from that?
Is that for me or Josh or
either one of you?
Happy to start. So preliminary efficacy in the context of this trial is really referring to the fact that all of the efficacy endpoints, so the things that we measured that look to efficacy, were all exploratory endpoints. So again, as we've been discussing, the study was not designed to answer the question, is ARM210 efficacious in this population? That was an exploratory endpoint. We looked at efficacy through a range of different tests, mostly physical therapy tests, one of which was the motor function measure 32, which is a 32-item questionnaire that looks at motor function, graded functional tests, so walking up and down 4 steps, walking 10 meters and moving from a sitting supine to standing position. And we also looked at the PROMIS fatigue questionnaire, and we focused on this based on the results of the prior NAC trial, which demonstrated that fatigue is a real issue that affects the RYL1 population. So those were the different efficacy endpoints that we looked at, in addition to muscle strength on physical exam.
All right, so can you talk about those results? So recognizing this is sort of exploratory, preliminary, What were your takeaways? What could you conclude based on, again, with the various caveats that we've mentioned? What sort of takeaways did you come up with on the efficacy of Rycals?
Yeah, so I think there were two particularly interesting takeaways. The first was the response on the promised fatigue. We saw a potential dose-dependent decrease in fatigue in this trial. And what I mean by that is at the low dose, we saw really no change in fatigue levels over the 1 month dosing period. However, in the high dose participants, 3 out of 4 demonstrated quite a significant decrease in fatigue over this period, albeit self-reported. The degree of change that we saw actually exceeded what was reported previously in terms of natural history change over time, and also what we know from a placebo arm of a prior study.
Okay. All right. So on the on this issue of fatigue, in the article, I think you just sort of made reference to this, 3 out of 4, that the recessive or biallelic patients did not show a decrease in fatigue. And in the article mentions that those patients may have had to have received a higher dose or treated for a longer period of time to have the same effect that was seen in the dominant or monoallelic patients. So I just want to put that out there. And then there was a question that we had directly from the community, which was, do you have any further details on the reasons behind the lack of efficacy on Rycals in patients with recessive mutations? So can you address that issue that it did seem that there was a decrease in fatigue in those patients with dominant or monoallelic mutations, whereas it didn't seem to have that same effect on the recessive patients? So can you sort of give us your thoughts on that?
Yeah, I would preface my response by saying that we are working with very small numbers here, and we can't draw any firm conclusions from this aspect of things. But the theoretical construct that's brought out in the paper is that the therapeutic target here is the ryanodine receptor, RYR1. And in certain recessive cases, we know that there's decreased expression of the RYR1 channel. And so it's theoretically possible that a higher dose or longer exposure to the product may be needed to see a therapeutic effect. But this really needs testing in a larger study, in a larger sample size, because again, there were only, I believe, 2 recessive individuals enrolled in this study.
And Mike, I might add that, as you probably know, many of the experts in this field who treat RYR1 patients have essentially told me that they consider these 2 different populations of the disease, the monoallelic versus the biallelic, because the symptom complex, the pattern, the muscle distribution are very different. So, so it's almost like 2 different diseases.
Yeah, and I think it's a great point. And I think for certain forms of the recessive, where there is a reduction in expression of the protein, there's just not as much RYR1 that perhaps may limit the efficacy of Rycal. We don't know that for sure, but that's certainly a possibility. I want to move a little bit towards next steps, but before we do that and shift gears to Armco and Dr. Marcantonio, a question probably for everyone here as it relates to what we were just talking about, the dosing. There was a suggestion in the paper that maybe a higher dose would have had a greater effect on those recessive patients, or perhaps even the dominant patients. Can you talk about the dosing that was used? The low dose was 120 milligrams. The high dose was 200. Where did those numbers come from? Could those numbers go higher? Was there a reason they didn't go higher in this trial? Could they be higher in subsequent trials?
Payam, you want to take a first shot?
Sure. I just want to mention something about the dominant and the recessive. And I know there's this dichotomy, but from a mechanistic standpoint, there's still a lot what we don't understand, especially because a lot of recessive patient or biallelic patients have 2 different mutations that may exert 2 different mechanisms at the same time. So I don't think it's as straightforward to say, for example, the recessive patients are not going to benefit from this drug, and the dominant patients are going to benefit. I think there are some dominant patients that may not benefit from this drug, and some recessives that could potentially benefit. But anyway, so I just wanted to mention that, at least based on my understanding of how this works. But to get to your question about the dose, so a lot of this in early development in phase 1 trials, you always kind of want to start at a low dose, assess the safety and the tolerability, and then keep going up on the dose until you either have side effects or you have gone as high as you can. And the sort of ceilings are in part determined by studies in animals. So the safeties, the toxicology studies that are done. And Gene knows all the details more than me, but obviously Rycal's went through this ARM 210 went through rigorous toxicology studies before FDA kind of had approved its use in the trial. And some of the dosing determinations was directed based on the side effects that were noted in the animal studies at super high doses. They give really high doses to the animals. And that kind of informs and is projected to the human study to have a margin of safety, usually tenfold safety from any kind of toxicity, equivalent toxicity in animals. So that's how this trial was designed, to get to the maximum possible dose that we can without going over those ceilings that were put into place by the regulatory bodies, in this case the FDA, which regulates the trials in the United States. Now that limitation or those calculations can potentially be revised by the regulatory bodies, or if you go to another regulatory body, like in Europe, they may have different calculations or different considerations. So the dose of the maximum dose of the drug can potentially be changed, especially since we didn't see any major adverse events. There is potential to potentially go higher to see whether you get even more efficacy. But again, in the United States, we were limited at this dose because of the FDA regulations.
Right. So Mike, just one thing, the 120 dose was picked to match an exact dose that we had done in healthy volunteers, so that we could measure the blood levels and compare patient doses versus the healthy volunteers. The 200, as Payam has pointed out, was below the safety margin set by the agency. However, you have to understand that when we put this study in for the agency, we had zero benefit. And so the benefit risk ratio was all risk, and there was no benefit. So as we accumulate some benefit and reduce the risk, the chances are that we should be able to go higher. And as Payam points out, that's very likely in other jurisdictions as well.
And when you say other jurisdictions, you're talking about different countries, correct?
Yes,
That's
yes,
beyond the FDA's purview, correct?
yes.
All right. Well, why don't we jump into that, Dr. Marcantonio? So the article concludes, Dr. Mohasel and Dr. Todd, in there, and you were a co-author as well, say that the findings from this study warrant further investigation in a randomized mass placebo-controlled trial. I assume you agree with that, but could you just sort of share your thoughts on, just generally speaking, next steps? So we've talked about the basis of Rycal, we've talked about the clinical trial and what it showed and maybe what it didn't show. You're the CEO of the company that is producing this drug. What is the future of Rycal's in, let's say, near term, medium term, long term?
Right. So there are several things we are doing near term. For example, we are going to dose in healthy volunteers higher than we had previously to help establish a higher safety margin, so that there is some room to potentially go higher. In addition, we currently have a phase 2 trial, which is a placebo-controlled trial in the Netherlands and in the Mayo Clinic in the cardiac condition, RYR2 mutations, and that's going well. It's a little bit tricky because we do that on top of another standard of care, but that is going well. And I think that will help build the case that the drug is benefiting patients with these kinds of mutations, albeit in a different RYR. In terms of RYR1, we plan on doing an endpoint study without drug initially. And the reason for that is it's not totally clear what is the best endpoint to study in RYR1 patients. Dr. Mohasel knows this very well. He was one of the people that suggested that we do such a study. And it really comes down to, we know that the PROMIS scale was successful, and we know that when Dr. Mahassel did a deltoid exam or shoulder abduction exam, he could see differences in strength that improved over time. So we are looking at ways to quantitatively measure. That was semi-quantitative because it's a subjective exam, if you will. We're looking for quantitative methods to measure and to decide which muscles to measure, because in most cases, as you know, proximal muscles closer to the body, are affected more than distal muscles in RYR1. The easiest muscles to measure are the distal muscles, such as a hand grip or a pinch, rather than measuring your hip flexor or your deltoid abduction. So we are looking at that, and we're running a study in London and in Paris and in Nijmegen in the Netherlands. Those are 3 sites that we're going to be running a study where we look at the endpoint. As that data accumulates, it helps us understand 2 things. It helps us understand how many times we have to do the test in order to get a stable measurement. Because as you all might have guessed, if I give you a barbell and tell you to curl it like this, the first time you do it, you might have a little bit of a flex of your muscle. But then if I teach you, well, do it this way, you might be able to lift more or go longer. And that education effect can contribute to a problem in the trial. So those are 2 things we're trying to do in order to run a placebo-controlled phase 2 trial, which at this point we hope to start by the end of this year. The other problem is we don't have a lot more drug. So we're making drug suitable to go to a higher dose in that trial.
All right, so Dr. Martineau, just to make sure everyone's clear, when you say an endpoint study, you're talking about people with RYR1 myopathy who are going to be assessed with these different tests of strength, but they're not receiving drug. Is that correct?
Not at this point. That's correct.
Okay, so in terms of a study, a phase 2 study that would involve people with RYR1 who would be receiving drugs, some receiving placebo, you were saying, and this is where most of the questions from the community are coming from. When do you think this would start? Where would it be? And probably the most common question we had is, how do I sign up? How would I know if I'm eligible? Can you just try to address some of those questions?
Sure. Well, at this point, because we plan on doing the study at a higher dose, and we know we can do that in the European Union and the UK, we plan to do it there and not in the US at this point. The other disadvantage of the US is that, or the advantage of the UK and the other places is, for example, in the Netherlands, the site in the Netherlands is essentially where almost all RYR1 patients are seen. So they're a captive audience, if you will. Similarly, the Institute of Myology in Paris collects a large number of patients from around France. We don't really quite have that here in the US. So between the dose limitation and the center limitations, at this point, it's better to do things in those countries, and we may add another 1 or 2 later. We'll have to see. In terms of the population, we do plan on doing the study in autosomal dominance or monoallelic patients initially as a follow-up of the efficacy here. Exactly how we approach the biallelics, though, is still open for question, and we're thinking about how to do that. The last point is we do hope to start it by the end of this year, but not much sooner than that.
And would these be adults, kids?
Yes,
both?
they would be adults at this point. It's possible we might extend it to teenagers who are similar size as adults. We haven't considered that yet. That will depend on what the investigators think. That would enable us to give a similar dose to all those patients. We don't have a huge amount of experience in children. We have the ability to go down to children as young as 2 years of age, in theory, but we have not measured blood levels or dosing in those younger children yet.
And would there be a certain neurological exam or certain in terms of the inclusion criteria as it relates to their functional deficits or weakness? Are there certain things that you're looking to that would be important criteria in terms of, do they have to have a certain level of weakness that or not too severely affected, or if they're too mild, it may be too hard to show a benefit? So what are you looking for?
So we expect people to have a deficit in a similar measure as you saw in the trial that Dr. Mahassel did, essentially a physical exam measurement of some important muscle group. And it doesn't have to be any one particular muscle group, but that's some clear weakness visible by physical exam in a muscle movement. In addition, we as you probably remember, we have a limit on how poor the diaphragm function in the lungs for the lungs are. So as long as the pulmonary function tests show that you are at least 50% of normal pulmonary function, you can be in the trial. But if you have less than that, the challenge is that that makes it difficult for us to avoid pulmonary adverse events that we can't adjudicate at this point easily. So that will be the main criteria, though, will be, as I said, the physical exam of these experts. These investigators are all experts, and they can assess the weakness. Now, many of the patients in the trial will actually be patients of the investigator as well. So they will have already some idea about what the nature of the weakness is in those trials.
Okay. So one of the most common questions we've gotten is, how would I sign up for this trial? So recognizing it's going to be people who are living in Europe, although we have people from Europe who are logged in right now, would they be able to sign up, or was it going to be those investigators reaching out to their patient population to pick and choose who would be included?
No, they can sign up, certainly if they can make it to the trial, because, you know, for example, if you are too far from a center and there are, let's say, 4 visits, which is likely, you have to be able to get there for 4 visits. Now, we pay for travel, but if it's very difficult to make it, or people are working and they can't get off that time, those are more the limiting factor. I think that it also will be useful in Europe and the UK that you reside in that country, simply because a lot of times the National Health Service or the insurance, the single payer country model is helpful. That doesn't mean you couldn't go from from France to the Netherlands, or if you were in Germany and you wanted to do the study in the Netherlands, you could do that. It's possible. I think even in the United States, you could theoretically be involved, but the tricky part there is health insurance and coverage and everything else. So that would make it unlikely, but there's no restrictions per se on geography.
Okay. Next question we have from the community, and this is asking people to look into a crystal ball, perhaps many years down the road. And I would open this up to everyone. And it relates to some of the things we've talked about already, but do you envision Rycal's potentially being efficacious in all patients with RYR1, or do you think it's going to be subpopulations based on, again, dominant versus recessive, or maybe their specific mutation and how it's affecting the functioning, the function and structure of the ryanodine receptor? Or any thoughts on that? Again, I know that's asking a lot.
Well,
and
one
probably-
population, I will say this, Mike, one population that we are not studying now, and we don't intend to, is the population that gets rhabdomyolysis, that they get leakiness of muscle components and overheat as a consequence of exercise. These individuals, especially Heinz Youngblood has told me, are often very strong, actually very athletic. They may benefit slightly from dantrolene, low-dose dantrolene, to help prevent that leak and reduce that overheating. We don't understand their molecular pathophysiology enough to try to a Rycal on that group right now. So in terms of the crystal ball, let Payam or Josh consider that one too, because I really, I don't know. My bias would be potentially yes, it should work in all patients who have weakness of some sort, but it's unclear.
Yeah.
I think it's the way I think about it is you need to kind of think about this a little bit in a step-by-step manner. So what can we show more definitively so that you can get the drug or any drug to the next step? So it may be easier to show that the drug works first in a particular subpopulation of the RYR1 community. And then if that works and get it forward, then it becomes easier to expand and test it in more and more individuals. And that also includes the issue of testing it in children, which we haven't really talked about. But usually, you want to test it, at the beginning when it's kind of risky. You want to give it to adults who are, older before giving it to a potentially vulnerable patient population, which is children, and then kind of adjust. And as time goes on, if everything is going well, then you kind of expand and give it to more and more individuals. So, I think it has potential to be efficacious for a large number of people, but with RYR1, but whether it's going to be helpful for everybody, that's probably too much to ask of any drug, and this one included.
Right. And
I think
Josh,
my one point that Paya made is that I like to say that drug development is like building a house, and you have to start out with a solid foundation before you get to the rest of the house, so it doesn't crumble on you. And this trial was one of those first steps in that building that foundation.
Josh, any thoughts?
Yeah, I was just going to actually build directly on what Dr. Mark Antonio said, that we're still really building the foundation of data to support these types of claims. So I think we're at the early stages, but it's great that the program's moving forward.
Yeah. All right, great. So another question we had was about children. Dr. Mahas, so I think briefly just referenced that, but it's fair to say that at least for the next short to medium term, next couple of years, at least, that the focus will be on adults. That's usually how things work in drug development. Is that right? And then at that point, if there's still good safety, good tolerability, maybe some efficacy, at that point, there'd be some contemplation of using this in children. Is that a fair?
Yes, that's correct. That's fair. And I think, you know, for example, in our cardiac indication where we've got some hints that it might be working, I think we might do children next potentially. But in general, most of the investigators have told us when some of these investigators treat both children and adults, they've said that, you know, the parents are going to be reticent to try something that they don't have an idea is likely to work. So it's also useful in that regard to have something that helps build confidence in the parents of the children that you're going to give doses to.
Great. So we're in the last few minutes here, and I think we're getting questions online right now, but I want to just try and maybe jump to a few additional questions in our remaining 12 minutes or so. This is sort of a basic question, but I think a good question. What is the hypothesis if Rycals are to be efficacious, and there's some indication that maybe in at least in some groups it is? What is that? What is the explanation for it? Is it its increase in muscle mass? Would it be just an improved functioning of the muscle cells? Can one of you try to address that question?
Sure, I could take that. So the leak in the muscle in the RYR is going to cause calcium that should be controlled for contraction to come out inappropriately. So a lot of calcium is behind the receptor, if you will, inside the rough endoplasmic reticulum or the sarcoplasmic reticulum. When the nerve tells you to contract a muscle, the channel opens, calcium flows out, and that drives the contraction, the sliding of the filaments that causes your strength. I like to refer that as ammunition for the gun that triggers your muscle to go. When you have a leak, you end up with less ammunition. So inevitably, when you tell the muscle to trigger, there's less ammunition and the muscle is weaker. The short-term effects we see are likely due to allowing the muscle to have more ammunition for that contraction. Size, not likely to change, but I guess Payam, you guys are more expert at what the effects of exercise could be in this population. But if you're stronger, you have a chance of having a better benefit of exercise. That's all I would say about that. So Payam, maybe you want to comment on that a little bit.
Yeah, I think you know, it comes to also a little bit building on what Gene is talking about is we know that there's a problem with the RYR1 channel, and that has a lot of downstream and long-term consequences. There's some short-term consequences of that calcium leak, and there are some long-term consequences of that. And I would expect that if you can address the underlying problem, as time goes on, you will address the short-term problems first and some of the long-term problems potentially later. And that muscle mass is probably one of the last things that's going to recover, whereas fatigue would be one of the first things that would potentially see a benefit. It is possible that muscle mass will not improve, but at least it will remain stable from that point onwards. And that's specifically true for some of the muscles that are completely remodeled at that point of the disease by non-muscle tissue. And we know that that happens to some extent in RYR1. just based on MRI studies. So we don't expect this drug, based on the way it works, to reprogram those non-muscle tissue, fatty tissue, connective tissue back to muscle, but at least it will prevent the loss of the remaining muscle. But the muscle that hasn't quite gone that way and is weak can potentially recover and get bigger for potentially even. But I'm really hand waving at this point about those expectations.
And those downstream effects you're talking about would be the effects or the sequela of prolonged calcium leak on some of the other components, things like mitochondria, actin, myosin, things like that.
Exactly.
So, okay. So the thought here is that based on what I'm hearing is that the Rycals work by one, improving the functioning of that ryanodine receptor, improving the overall functioning of that excitation, contraction, coupling, the actual contraction of the muscle. But then, as Dr. Mohas was saying, some of those downstream effects may also be mitigated if you can improve or reduce that calcium leak with Rycals. And
So
muscle
Mike,
mass.
one quick comment about dantrolene versus a Rycal, because I know that was posed.
Yeah, that was
And
my
people
next
are
question,
saying, well,
actually.
dantrolene closes the channel, so why wouldn't it work the same way? I can't completely explain all the mechanistic differences. Some of them will be coming out from some structural work by our founder, Andy. But the bottom line is there's many papers that show that dantrolene actually worsens muscle weakness in animal models and in patients. It depends a little bit on the dose, but it does close the channel, but it doesn't quite allow normal function either. So it's not as useful as a Rycal, which does allow for the channel to be closed, but also regain its normal function.
All right. So just a couple more questions here. The things that we're seeing the most commonly asked is, and again, simple question, but probably not answerable, or at least not a simple answer, is when would this drug come to market? I know that, yeah, it's probably impossible to answer, but can you maybe address what are the things that would be determinative of when it would come to market? What would slow it down? What would speed it up? What would prevent it from ever happening? Can you just talk about that process a
Well,
little
I'm not
bit?
going to talk about how it prevent it from ever happening. There's too many ways in which a drug can die.
Right.
And I'm pretty much expert at that because that used to be my job, was to kill drugs. So, but essentially in a rare disease like this, very compelling placebo-controlled efficacy is likely to spur a lot of interaction with the agency through either breakthrough or other kinds of special status, which then may allow a rapid or preliminary approval. So if there is a very strong response in the phase 2 that we plan, with a strong efficacious signal that's unequivocal, then it is likely that we would be having rapid conversations with the FDA as to what it would take to actually get a preliminary approval. Right now, it's hopeless for us to have those discussions. We're down in the basement. We're not up on the next level yet of the house, if you will. So, but that's the best bet for the compound to be approved for the orphan diseases more rapidly, hopefully within a year or two after a very nice result in a phase 2 trial. I don't think that this kind of a drug for these things needs a full dose ranging, phase 2B, or a full phase 3 trial that typically, or certainly not a duplicative phase 3 trial, as many other indications would need. I think strong placebo-controlled evidence will drive the agency to help us in an iterative way as to how to register. That's the best I can say in terms of the hope. What can you guys do to help us? What can the patient
That was the
population
next question.
besides joining the trial, obviously, which is one thing, two things that the patient population can help us with. They can help us give feedback on the endpoints we're using as to how relevant it is to their lives, because that's the kind of impact the agencies want to hear. And then if we do get involved with the agency and we get to an advisory committee or whatever, I'm going to ask you to put your tie on again and come and testify before them to talk about the importance of the drug in the disease. So those are the two places where it can. But it is useful for the patient population to give us feedback on endpoints if they've been enrolled in trial, or they just read about those endpoints and say, stair climbing is really a big problem for me. If you can show me that your drug is improving that, that's going to really mean a lot to me. That sort of feedback is very important to the agencies these days.
Great. Thank you. That was really, really helpful. We're essentially running out of time. Are there anything else either from Dr. Marcantonio, Dr. Todd, or Dr. Mahassel? Anything else you think our community should know or should be aware of, or anything else we
I
can be
think,
doing?
Mike, I'd like to say one other thing, that I know there was some concern about what would happen, how can we commercialize, how can we afford to do orphan diseases? Orphan diseases are not impossible to do. And that's one thing. And the second thing is, what happens if we get bought by a big pharmaceutical company? Will we lose the RYR1 indication? I don't have a crystal ball there, but all I can say is that ARMGO is highly committed to both mutation diseases in RYR1 and RYR2. This is what we are doing right now. Where the data leads us is another story, but that's our commitment.
Well, thank you. That's great to hear. I really appreciate that. Dr. Mahassel, Dr. Todd, anything else? Any last final comments?
I do have one other comment for these two gentlemen. You talked about how difficult it is to do a trial. This trial was done during the pandemic. It was supposed to start in March 2020. Obviously, nothing on the East Coast of the United States other than bad stuff happened in March 2020. It was delayed a long time. Then we had to wait for people to be vaccinated. PIAM had to deal with people who came into the trial we thought were COVID negative and were COVID positive. So it was really, really a tour de force to get this done during the pandemic. And these two guys need all that credit.
Well, I agree with that wholeheartedly. It was an amazing thing to see it get done. The work leading up to it, the pandemic, some of the issues that we had even prior to the pandemic. If people, we could have done 3 hours just on how we got this trial done. I think it should be made into a movie. I think George Clooney should play Dr. Mahassel. Brad Pitt, Dr. Todd, I don't know. But it really was an amazing thing to see happen. And it was, as I said earlier, it was a group effort, and it couldn't have happened. You know, it required advocacy, it required the expertise from all you guys, industry, NIH, academia, the work done at Columbia by Dr. Mark. So it really was an amazing thing. And I think it's a prescription for other therapeutic endeavors that we pursue. Last but not least, I want to thank Lena, our administrative assistant. I told her there's always an IT snafu, and as far as I can tell, with 2 minutes left, we've had none. So nice work, Lena. Thank you very much.
Mike, I just wanted to mention something. So, even though Josh, did, really 70-80% of the work behind the scenes, we had a large team. We had study coordinators, nurses, who really drove the project forward. It wasn't just the two of us. There's like a whole army of people, and most of them are acknowledged in the paper as either as co-authors or in the acknowledgement section. So I can't, we can't, both of us can't take the credit for this, really. I think, and as you mentioned, I think the partnership has been very rewarding. Again, at the time I was at the NIH, so part of federal government, with ARMGO industry, the foundation, sort of these different groups and academia, each one with their own interests and expertise sort of coming together to move the mission forward. And I think that's really the framework of any future trial for this disease. So happy to be a part of it.
Yeah, now you're an RYR1 expert, whether you like it or not. We got you. All right. I think that's it. Thank you all for doing this. I really appreciate your time, all your efforts. The turnout we've got on this webinar has exceeded anything we've ever done before. So there's a tremendous amount of interest. And thank you for those people watching. Please reach out to us if you have any other follow-up questions or comments or concerns. But thank you for participating. And everyone have a great evening. Thank you very much.
Thank
Get her,
you.
Mike.
Yeah.